Hello and welcome to the second database activity relating to the International Baccalaureate Diploma Programme's biology course for topic 3, genetics. In this short screencast, I'm going to demonstrate how you can use the Ensemble Genomes online database to address the following 3 assessment statements from the syllabus, notably Application 2 in topic 3.1, which is comparing the number of genes in humans with other species, and then the second application in topic 3.2, which is comparison of the genome size in the T2 phage, Escherichia coli, Drosophila melanogaster, Homo sapiens, and Paris japonica. And then third application in 3.2, which is comparison of diploid chromosome numbers in the following species. I would like to highlight again that I have actually used 2 resources to help me develop this screencast. I've actually adapted the protocols given in the Allen-Mindorf biology textbook that is a course companion for the International Baccalaureate Diploma course in biology, as well as the biology teacher resources from the InThinking website there. So one thing that I give the students then will be this table that needs to be completed. Again, I have adapted this directly from Allen-Mindorf and combined it. They actually supplied the numbers from that in that textbook for these particular areas. I thought I'd leave these in as they might be particularly challenging. Some of these species are not included in the Ensemble database, although through a bit of Google searching you can actually find, for example, the number of genes for the T2 phage, obviously not the diploid number. However, on the side of being keeping things simple, I just wanted to look at one specific database which can be used to find the following information. So the number of genes for the following species, wherever there is a white box, that information can be filled in. So first thing we need to do is run to our Ensemble genome webpage. So you can Google Ensemble genomes. You could just type this into your browser. Now you'll notice that on the top right here we have these different tabs. So we have the database for the bacterial genomes, the protist genomes, fungi genomes, plant genomes, metazoan genomes, and then the vertebrates. And I'm just going to show you how to just search for one of these particular species, Homo sapiens. So wise man, modern man is a vertebrate. So I'm going to go to the vertebrate genome database, and here we have our Ensemble genome database. Now we can browse a genome. Popular genomes is here is human, but you can also select a species down here. So I'm going to go to the human via this route just to demonstrate that you can use this. You actually have a list of different species that you can see in this database. Okay, so we've got fish and amphibians, birds, reptiles, and mammals, and we're obviously going to go to the human and click through. So now we're on the information about the human, and here for all the species we see we have view carrier type. So I can click on this, it goes to go to human carrier type, click here, and there we have it. We have a carrier type image. Okay, so this will allow you, by looking at this, you can actually work out the diploid number of a species, and then if we click down through the database here, we can see that we actually have the information that we need for this exercise. So our base pairs, this is the total number of base pairs within this genome. So this is the genome size, and in the table from Allen-Mindorf, this is given in million per million base pairs. So a short calculation can let you work this one out. So for genome size, we need to look at the base pairs, and then for the number of genes, we're going to look at the coding genes here. So how many actual coding genes are there in this specific database? And that's simple. That's really it. We can use that database then by going back to the over the top here, to look at the bacteria or fungi, whatever it might be, the particular species that we want to look at. So Escherichia coli will be in the bacterial area, Drosophila melanogaster is in the metazoan area, and so on and so forth. Thanks for watching. I hope that has been fairly straightforward. If you like what you see, you can follow me on Twitter at WRPVincent.